Tens of millions of mice are used for all kinds of scientific research every year worldwide. They are typically kept in small, empty cages not much bigger than a Kleenex box. Because of this, laboratory mice engage in abnormal, repetitive behaviors like constantly backflipping, spinning in circles, and gnawing on the bars of their cage. Well, these are called stereotypic behaviors, and they are common in zoo, farm, and laboratory animals living in cages that are too small and too empty. If instead animals are kept in large, complex cages that allow them to engage in their natural behaviors, they're not stereotypic. So why do captive animals do these things? They may be bored or trying to escape, or they may even be a way of coping with stress. But what if I told you they could indicate brain dysfunction? This was the focus of my master's thesis. One common hypothesis is that, much like humans with autism or obsessive compulsive disorder, which also involve repetitive behaviors, stereotypic animals may have abnormal functioning in the brain system that both activates and suppresses behavior, the basal ganglia. When this system malfunctions, individuals have trouble disengaging from a behavior and end up repeating the same thing over and over again. To find out if this is true for stereotypic lab mice, I raised my mice in typical lab cages or large complex cages full of things to do and compared their behavior. It was no surprise that the conventionally housed mice were severely stereotypic, spending most of their day just running around and around their cage. Next, I asked whether the type of cage affected their basal ganglia function, and if it did, could these brain changes explain why some mice are more stereotypic than others? To answer this, I used a chemical test that showed me which brain regions were more or less activated based on how dark they appear. I found preliminary evidence that highly stereotypic mice, one part of the basal ganglia was different, the nucleus accumbens, which is considered the reward center of the brain. This suggests that some individuals may have enhanced motivation to engage in stereotypic behaviors, finding them rewarding, perhaps even addictive. But my work is not done because conventional caging did not affect this system, leaving the brain mechanism that causes stereotypic behaviors a mystery. So now I have to look in other brain regions. But why continue? Because if typical laboratory cages cause brain dysfunction, this would obviously have significant ethical implications for laboratory mice, and it could even have serious implications for science itself, given that most animal experiments fail to yield results that successfully translate to human medicine. And so I wonder, could we be contributing to this problem by using animal models who are abnormal? Now that really would be insane.